Okay. I guess everybody is okay then. Fine, no issues. Okay, so once again, guys, what I want is just have a look at how mitosis has proceeded. Okay, so let's just once again go back. Okay, so see, we had said what is mitosis? Mitosis is going to be, it is divided into karyokinesis and cytokinesis. Karyokinesis further divided into prophase, metaphase, anaphase, telophase. And then we are going to have cytokinesis, that is going to be division of cytoplasm. Guys, today we are going to talk about your meiosis. And in meiosis, we are going to see something different that is going to happen. Okay, because remember, guys, here, what is going to happen, you are going to see division taking place twice. Okay, how? What happens out over there? Let's try to see. Okay, so guys, when we talk about the this thing, okay, one second for Zoe. See, this is mitotic apparatus, the chromosomal fibers and the interpolar fibers. Okay, and here this is the interchromosomal or the interzonal fibers. Okay, so guys, let's try to see what is meiosis, where is it going to happen, what really goes out over there. Okay, so remember when we talked about your mitosis, guys, where did mitosis occur? Mitosis occurred in all your somatic cells, and that is why we said it was your somatic cell division. But guys, when we talk about your meiosis, where does meiosis take place? Meiosis is going to take place only and only in your germ cells, meaning in your the testes or the ovaries. And what are this going to give rise to? They are going to give rise to your gametes, that is the sperm and the ovum. Okay, so that is what is going to happen. But guys, mitosis, who was the one who gave us mitosis? Quickly chat and answers, guys. Who gave us mitosis? Quickly. Very nice, Zoe. Very nice. I've received only answer from Zoe. What about the others? Guys, quickly. Akanksha, Darshanita, quickly. Kanaka, who gave us mitosis? We've done this thing. Come on. Who was he? He was Walter Fleming. Okay, he was Walter Fleming. But now, guys, when we talk about meiosis, who gave us meiosis? Meiosis was given by a set of scientists. Who is it? We have Van Beneden. Okay, so we have first we have Van Beneden. Okay, second we have Farmer. Van Beneden, we have Farmer, we have Moore, we have Winnie Waters, Winnie Waters, and lastly, guys, we are going to have Edward Strasburger. Edward Strasburger. Okay, so uh, these are some of the people who were associated with meiosis. Now, guys, if you remember, we have talked about this Edward Strasburger. Okay, when Walter Fleming was working on mitosis, at that time he had no idea what was really happening. So what happened was he knew Edward Strasburger, and when he discussed this thing, Edward Strasburger said that, oh, I am working on a similar kind of a concept which is called as meiosis, and in my thing, this thing is happening. Okay, so immediately Walter Fleming went back home and then he tried to find out that, oh, what is happening. So this guy, he had given assistance in mitosis as well. And guys, there was one more guy which is involved whom we generally don't talk about. His name is August Weismann. Okay, so just keep this thing in mind. His name is August Weismann. Okay, so guys, in your meiosis, these people are included. Okay, all these people are included. Keep this thing in mind. Okay, but once again, like I said, where does this takes place? This takes place in your reproductive cells, or we say it is going to take place in your germ cells. Okay, now guys, always remember, reproductive cells are also called as germ cells. Okay, now sometimes what happens now in your NEET exams, okay, they will ask you identify whether the following statements are true or false, and then one of the statement would be meiosis take place in the germ cell, true or false. And then suddenly you think germ, अरे germ मिले तो bacteria, germ बोले तो it can be some kind of a disease causing agent. So in that, I don't think so. Mitosis should take place. Meiosis will not occur. No, guys. When they are talking about your germ cells, they are actually referring to your testes and the ovaries. Okay, so keep this thing in mind. Germ meaning it is germinal epithelium. That is what they are talking about. Okay, and one second. So where are we going to talk about? So see, in the case of your man and in the case of your women, okay, male or female. So in the case of male, where is it going to take place? It is going to take place in your testes. It takes place in the testes, and what is it going to give rise to, guys? This is going to give rise to our sperms. And when we talk about the women, this is going to take place in the ovaries. And what is this going to give rise to? This is going to give rise to ovum. Now guys, always remember, you have three different terms. One is ovum, one is ovule, and one is egg. Okay, now remember, egg is a general term you can use anywhere. But when we talk about ovum, guys, ovum is the one which is used in animals. And ovule is the one which is going to be used in your plants. Okay, so this is one thing that you always need to remember. But now, guys, let's try to see what exactly is happening in your meiosis. But before we talk about meiosis, guys, once again, we'll have to see what was happening in mitosis and what really happened so we can compare it with meiosis. So let me just first give you the cell diagram, okay, the cell diagram for your mitosis. So, guys, what had we said? In your mitosis, we started off with a cell. This cell was 2n, right? And now we said after your mitosis, what is going to happen? This cell is now going to divide into your two more cells, right? And guys, each cell is going to be what? Is it going to be 2n or is it going to be n? Guys, quickly tell me quickly. Yes, very nice, Zoe. Yes, others, I want others to answer. Come on. Yes, very nice, very nice, very nice. Okay, so guys, you are going to see that each cell out over here was 2n, 2n only. Okay, now why? Because this kind of division, this was your equational division. Mitosis was your equational division. And in equational division, you are going to see that, oh, you are going to have the same set of chromosomes as the parent cell. But now, when we talk about meiosis, guys, now let's try to see how are things going to change a little bit. Okay, so when we talk about meiosis, guys, see, this was only one single division, right? Meiosis, I said, division is going to happen twice. How? Let's try to see. So, guys, first of all, you will always start with 2n only. This is your body cell. Now, whether it is your germinal cell, whether it is your cells of the testes, ovaries, it will always be 2n because your each and every cell of the body is going to be 2n, except for the sperm and the ovum. Okay, so now what happens is this cell, this is now first going to undergo your meiosis 1. Okay, so this now undergoes meiosis 1. Okay, now as it undergoes meiosis 1, guys, what is going to happen? You are going to get two new cells. Okay, but now what is the difference, guys? The difference is these two new cells that are formed, they are going to be n and n. Okay. So what has happened, guys? See, you were deployed out over here, and suddenly the two new cells that you have got, you are going to be haploid. So can I say the number of chromosomes has decreased out over here? Right? Because the number of chromosomes have decreased, guys, what are we going to call this thing? This kind of thing, this is called as your reductional division. Okay? So the first meiosis one itself is your reductional division. Okay, but now once you have got these two cells, now these two cells, they are going to undergo the second division. And what is that? They are going to undergo meiosis 2. But guys, meiosis 2 is going to be exactly same as your mitosis. There is no change. So this meiosis 2, okay, this is going to happen. But guys, what did I say? Meiosis 2 is going to be equivaginal division, right? Meiosis 2, I mean, it is going to be same as your mitosis, so it is going to be equational division. So from one cell, how many cells are you going to get? You are going to get two new cells, right? So each cell is going to give two new cells like this. And now what is going to happen, guys? Now you are going to see that, oh, each cell is going to receive the same number of chromosomes as this parent cell. Now this is going to be the parent cell for this next division. Okay, so ultimately, guys, what are you going to get? You are going to get from one parent cell. How many cells have we got at the end? We have got four daughter cells. But then what is the difference, guys? The difference is going to be that now here, in the case of your mitosis, the chromosome number in the new cells had remained the same as your parent. But here, when we get four new cells out over here, the chromosome number is reduced to half. And that is the reason, guys, that is the reason we are going to call meiosis as reductional division. Okay, everybody all right with this thing? Sure, no problem. Okay, so always remember whenever they ask you, okay, they will always ask you what is meiosis. So you can always say meiosis is nothing but it is reductional. This is nothing but this is going to be reductional and equational division, whereas mitosis was only your equational division. Okay, but now if we're talking about meiosis, okay, once again, I talked about plants also. Like, in plants we are going to have ovules. So let me just quickly show it to you in plants also what will happen. So let's say if we are talking about plants, then in the case of plants, we are going to have the male part. Now, who is the male part? It is going to be the stamen. That is nothing but your androecium. And who is going to be the female part? It is going to be the gynoecium. Or we are going to say that this is going to be made up of your carpel, or it is pistil. And these stamens, they are going to have the anther. In the anther, we are going to encounter pollen grains. Okay, so pollen grains, they are going to be the product of meiosis in your plants. And in the case of carpel, we are going to see that we are going to have ovule. Okay, so this is what is going to happen. Just, I'm putting it down, so that if by any chance you feel like, okay, I want to know what happens in plants, so that is why. Okay, but now, guys, we are talking about meiosis. But where does meiosis occur? Like, when we talked about the cell cycle, we had said that, oh, in the cell cycle, we had mitosis, and then we had G1 as G2 phase, right? So, where is meiosis going to fit then? Okay, chalo. So, let's try to see where is meiosis going to fit in that. Okay, chalo. So, guys, once again, let me just show you. Okay, so we had absolutely right, Kanaka. Absolutely right. Very nice answer. Very good. Very good, Kanaka. Okay, so guys, when we started off, okay, so we had said that, oh, here on the top, we said we are going to have mitosis. Then we said we are going to have G1. Then we said, okay, we have G2 with S, and then we had G2. Once again, from here, we had the G0, right? Guys, now I want you to tell me quickly, okay, guys, G1 as G2, all of these together, guys, what did we call them? G1 as G2, all of them. Very nice, Zoe. Very nice, Kanaka. But I want answer from the others as well. Arshia has not even not responded. Arshia, are you there with us? Yes. Arshia, answer. Sorry. Arshia, I want answer from you. Arshia, do you know the answer? Sachi. Sachi has also not answered. Sachi. Sachi, are you there with us? Okay, anyways. So, yes, what is this? Yes, all those who have answered, very nice, guys. This is nothing but your interface. Okay, now what is going to happen, guys? In the case of your meiosis, okay, exactly the same thing. Okay, so let's say this is the entire same thing out over here. What is the change, guys? The change is only, instead of your mitosis, now we are going to just write down meiosis. That is the only change, guys. The rest, everything is going to remain the same. So we have G1. From the G1 we are going to have the G0, let's say G1, the S, the G2 phase, and once again all of this is nothing but your interface. Right? So this is exactly how it is going to happen. Okay, but now guys, now slowly what we have to do, we have to go and we have to see what exactly is going to happen in meiosis. Okay, so we said chalo either this is the entire cell cycle, this is where meiosis is going to take place, but what exactly is going to happen out over there, we need to find that out. Okay, so let's see what exactly is going to happen. So guys, once again, let me just give you a brief recap. Okay, brief recap what had happened. Okay, so we said we had mitosis. Okay, and mitosis, we said this is divided into your two parts. Okay, what were the two parts? We have karyokinesis and we had cytokinesis, right? Once again, karyokinesis, we said this is divided into prophase, metaphase, anaphase, and telophase, right? And then finally we followed cytokinesis. Guys, this was our plan for mitosis. Okay, but now when we talk about meiosis, guys, meiosis, I told you this is going to take place in two phases. So let's try to see what is going to happen. Okay, chalo. So meiosis is completed in two parts. So once again, we are going to have meiosis 1 and we are going to have meiosis 2. Okay, meiosis 1 and meiosis 2. Now, guys, what will happen? The cell is first going to enter into meiosis, then it is going to enter into your meiosis 1. After it finishes off this meiosis 1, then there may be a small gap meaning a resting period between meiosis 1 and meiosis 2, during which the cell is not going to be active at all, meaning it is not going to be doing anything, it is just going to be resting. What is that resting phase, guys? That resting phase is called as interkinesis. The resting phase is called as interkinesis. And guys, this is not a necessity. This is not a necessity. It may or may not be there. Okay, it may or may not be there. There is not a necessity that, okay, it has to be, it is a compulsion. No, it is not of that sort, guys. Okay, but now what is going to happen? Chalo, let's try to see. The cell is going to undergo meiosis. In meiosis, it's first going to undergo meiosis 1. But now, the moment it undergoes meiosis 1, guys, once again, what is she going to undergo? She is going to undergo two phases, just like your mitosis. So we are going to have karyokinesis. Okay, and we are going to have cytokinesis. But guys, see, in your meiosis 2 also there will be karyokinesis, there will be cytokinesis, so we will get confused. So what are we going to say? We are going to say that, oh, this is karyokinesis 1 and this is karyokinesis 1, karyokinesis 1 and cytokinesis 1. So meiosis 1, the cell is going to have karyokinesis 1 and cytokinesis 1. But now let's say the cell has entered karyokinesis 1. Now, karyokinesis 1, guys, this is further going to be divided into four more parts. See, because karyokinesis was divided into prophase, metaphase, anaphase, telophase, right? But now, once again, what are we going to divide it? Prophase 1, metaphase 1, anaphase 1, telophase 1. And then the cell is going to enter into cytokinesis. Okay, fine. But now, guys, the next thing that we are going to see is that even if it enters into your karyokinesis, then it enters into prophase. Immediately, it is not going to enter into my metaphase 1. In fact, the prophase 1, there are going to be 5 different stages that are going to happen out over here. So let's try to see what are those five different stages, guys. So the five different stages are, first one, we have leptotene. What is going to happen in all this thing? I'll tell you. Leptotene, this is also known by another name that is called as leptonema. Leptonema. Okay. Second one, after leptotene, we have zygotene. Zygotene is also known as zygonema. Third, then it is going to enter into your pachytene. Okay. So leptotene, zygotene, pachytene. This is also known as pachinima. Okay. Then we have your diplotene and diakhinesis. Okay. Diplotene and finally diakhinesis, like this. Okay. And once again, your diplotene, we can call this thing as diplonema. Okay. Diplotene. Teen means what, guys? Always remember, whenever we talk about teen means stage. Nema means thread like structures. Okay. So this thing. But now the question arises, guys, how are you going to remember these five stages? Because you have to remember them in order only. So how do I remember this thing? Okay, I remember this as LYPDD. Okay, LYPDT, leptotene, zygotene, so ZY, so zygotene, pachytene, diplotene, and diakhinesis. But then some people may get confused out over here. So for you guys, you remember this as lazy pad cis. Lazy pad cis. Okay, doesn't make sense, but leptotene, zygotene, pachytene, diplotene, and diakhinesis. Okay, because both of them were DD, so I have taken cis from here. So you just remember this as lazy pad cis. In your NEET exam, guys, they will ask you always, sometimes to arrange them in the right chronological order, or they will ask you what happens in each of the stages. Okay, so that is why you need to know this thing in proper order. Okay, so now guys, let's say the cell is wanting to undergo meiosis. What is going to happen? Okay, so guys, please pay attention. Okay, everyone. Okay, so the cell has first entered into meiosis. The moment it enters into meiosis, where is it going to go? It is going to go into meiosis 1. The moment it enters into meiosis 1, it is first going to enter into karyokinesis 1. After it enters into karyokinesis 1, then it is going to enter into your prophase 1. From prophase 1, now what is going to happen? First, it is going to enter into leptotene. Leptotene, then it is going to enter zygotene, zygotene, pachytene, diplotene, diakhinesis. This is where, guys, prophase 1 is going to end here. But now after diakhinesis, now what is going to happen? Then the cell is going to enter into your metaphase 1, anaphase 1, telophase 1. Then your karyokinesis is going to end. Then the cell is going to enter into your cytokinesis 1. This is where from one cell, okay, now see, this is the end of cytokinesis. So at this time, you are going to see that this first division, this first division has taken place. Okay, then the cell will enter into meiosis 2. Now, after this, once again, we said, oh, we are going to enter your interkinesis. Interkinesis may be there or it may not be there. Okay, and then it is going to enter into your meiosis 2. But now let's try to see what is going to happen in meiosis 2. Okay, so let me just remove these arrows off. One moment, I'm just removing these arrows. Okay, so now guys, after all this thing, after your interkinesis, which I said it may or may not be there, the cell has entered into meiosis 2. Okay, but now as the cell enters meiosis 2, once again it is going to enter into karyokinesis, right? But now here we are going to have karyokinesis 2 and cytokinesis 2. But now once again, karyokinesis too. This is further going to be divided into prophase, metaphase, anaphase, telophase, right? So once again, you are going to have the same kind of situation. So we are going to have prophase, but now here this is going to be prophase 2, metaphase, metaphase 2, anaphase, anaphase 2, and finally we are going to have your telophase, telophase 2. This is going to be exactly the same. But now, guys, when we talked about your meiosis 1, we said, oh, in your prophase 1, we are going to have these 5 stages. Are these same stages going to be repeated in your prophase 2? Answer is no. Here, you are just going to have only this thing, guys. Okay, so this is the entire map. This is the entire map for your meiosis. Okay, so guys, once again, I want you to see this thing. Just see if you are okay with this, because the same like mitosis, right? Yes, because it is exactly same as mitosis. Yes. Okay, guys, just have a look. Just see if anybody has any doubt with this plan. Once again, P1, M1, A1, T1, P2, M2, A2, T2. These are nothing but prophase, metaphase, anaphase, telophase only. Okay, just guys go through this once. Sorry. Okay, and always remember, guys, whenever you do this thing, okay, the first thing is you need to know this map. Okay, if you don't know this map, then you are going to be confused, like where what is happening, how it is happening, and why is it happening. Okay, is everybody okay with this? Guys, give me a thumbs up if you a thumbs up. nahi, abhi, I at least say yes, yes. Okay, 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 okay. Okay, so guys, now what are we going to do is we are going to enter, okay, into your this thing. We are going to enter into your actual meiosis, and let's try to figure out what exactly is going to happen out over there, guys. Okay, so once again, we have to see the cell diagram, just like how we had seen in your mitosis, like what were the things that were happening. Okay, exactly the same thing we are going to see out over here. Okay, chalo. So, guys, first thing is once again, before we move ahead, we always need to know how is the interface cell going to look like. Okay, so once again, exactly the same thing like how we had done it in your this thing. Okay, so always remember first we are always going to have interface cell. Okay, chalo. So, guys, once again in your interface cell, what have we said? Okay, we are going to have your the cell membrane. Okay, so the cell membrane is going to be intact. Then we said, okay, we are going to have your nucleus. Okay, we are going to have your nucleolus. Okay, fine. Then let's say we said we have your chromatin fiber, which is going to be like this, and we are going to have your centrosome. Okay, like this. Now, guys, this cell is wishing to undergo meiosis. That means it is willing to undergo your cell division. Can anybody tell me what is the first step that is going to happen? I told you while we did mitosis, guys, I had told you this thing. So, can anybody tell me? what will happen first? The first initial step, Jeevan? No, 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 no. My question is like, the cell has entered into your mitosis. Interface ho gaya. Interface is done. I am entering into your meiosis. Okay, just like, or you can say mitosis or meiosis, both. What is the first change that you are going to observe in the cell when the cell will undergo your cell division? The chromosome splits. No, you're just right out over there. The centrosome splits. Guys, always remember, when the cell is entering your cell division, no, no, not centromere, centrosome. No, 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 no. See, centromere. See, where is the centromere? See, we have the chromosome like this, right? And the chromosome right in the center, we have this gola gola. This is going to be your centromere. That was your centromere. Okay. So we are not talking about centromere or your chromosome, the centrosome, this one. Okay, the centrosome has to split, guys. Okay, so always remember that. Okay, anyways, chalo. So now, guys, how are we going to go? Now, this interface cell, no problem, no problem. Okay, sometimes it happens. थोड़ा सा इधर उधर confusion हो जाता है. No issues. Okay, now this interface cell. Now, see, guys, the interface cell, where is it going to land up? It is going to land up in meiosis. Meiosis one. Okay, I did not talk about the other part. So, guys, after your cytokinesis one, then what is going to happen? The cell, we said it is going to enter your interkinesis. Then interkinesis, then it is going to enter into your meiosis two. From your meiosis two, then karyokinesis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis 2. After it does cytokinesis 2, then it is going to give rise to four cells, okay, from one single cell. Okay, sure. But now let's try to see this interface cell. Now, what is this going to do? This is going to enter into your meiosis. Okay, and in your meiosis, now what is going to happen? This is going to enter into meiosis one. In meiosis one, this is going to enter into karyokinesis one. Karyokinesis one, then it is going to enter into prophase one. And after your prophase one, now it is going to enter into the first step of prophase one, that is leptotene. Okay, so this is now going to enter into your leptotene. Okay, now, guys, what exactly is going to happen in leptotene? Let's try to see. So, okay, so we are going to see that our cell membrane, it is going to be intact. It is as it is. Okay, the nuclear membrane, it is going to be intact. Okay, it is once again as it is. The nucleolus is also going to be intact as it is. But, guys, when the cell is about to divide, what did I tell you? The first thing, the centrosome is going to split and the centrioles are going to be formed. So once again you will see that, oh, the centrioles have come up. And now, what is the job of the centrioles, guys? We had said that the centrioles, they will start moving towards the opposite poles. But then, while it is doing this thing, at the same time, what is going to happen? They're still going to be attached together with the help of spindle fibers. This is always going to be there, right? So this is how it is going to work first. But now, leptos. Now, what does mean leptos means? Leptos means thin, thread-like. Teen means stage. Leptos means thin, thread-like. Okay. But now, who is going to be thin thread-like in this case? Leptotene. It is going to be your chromosomes. So what happens is you are going to see that the chromatin fiber which was present out over there, now this starts condensing and it gives rise to chromosomes. And how are these chromosomes? See, we are having two sets of chromosomes. So slowly, you will see that, oh, we have one chromosome from the mother and let's say the similar type of chromosome from the father. Let's say one more. You are going to have one chromosome from the mother. Let's say the similar type of chromosome from the father. like this. So you are going to see a situation that the chromosomes, they become thin thread like structures. That's it. That is what we need to know about a leptoteen. Okay, so guys, one thing that you're going to see, that the chromosomes, whatever the chromosomes were there, the chromosomes appear thin thread like structures. The chromosomes, they appear thin thread like structures. And now, what is the most important thing, guys? When this is going to happen, the centrosome splits. That's it. Okay, you have to keep this thing in mind. Okay, but now what is going to happen? See, I told you these chromosomes, one is from mother, one is from father, one is from the mother, one is from father. Okay, fine. Let's try to see what is going to happen. Now, after the leptotin, guys, we said, what is the next stage? The next stage is going to be zygotine. Okay, but now what is zygotine? Zygo means pairing. Okay, zygo means pairing, and teen is stage. Okay, so zygotine or zygonema, this is pairing stage. Now, who's going to pair out over here? Guys, these chromosomes which we had got from the mother and father, who are similar to one another, they are going to pair up against one another. So now let me just show you what exactly is happening. So once again, guys, in the next case, cell membrane is as it is. The nuclear membrane is also going to be as it is. There is no change. The nucleolus is also as it is. The centrioles, they are still moving away from one another, trying to reach the opposite poles. Once again, even if they are doing this, they're still going to be held together with the help of our spindle fibers. Okay, but now what happens? You are going to see that these chromosomes which were there, the thin thread like chromosomes, they are going to come up together, and now they are going to be arranged besides one another like this. Guys, this stage, this stage where they are going to be arranged against one another, parallel to one another like this is known as synapses. We say that these are going to be arranged in a zipper like fashion. So see, one chromosome is like this, the other chromosome is like this. If you just add a zipper out over here, and you can pull it up or pull it down, however, so this appears to be like a zipper like fashion. That is why we say that the chromosomes, they are arranged in a zipper like fashion. Okay, getting this thing. So what is going to happen in this stage, guys? And that's it. That is what we need to know about your zygotine. Okay, so we are going to say that in your zygotine you are going to have pairing of homologous chromosomes, pairing of homologous chromosomes in a zipper like fashion. And this is nothing but, we call this thing as synapses. Okay, but now I have used the term homologous chromosome. Okay, so what are these homologous chromosomes? Let's understand this. Uh-huh. Okay, so guys, see, how many chromosomes do we have? We have 46 chromosomes in our body. But what have we said? 46 chromosomes is actually going to be two pair. Like, it is basically one set of chromosome is going to come from the mother, one set of chromosome is going to come from the father, right? So that is how we are going to have 23 pairs, meaning two pairs, meaning two sets of chromosomes, right? So let's try to see how is it going to happen. Okay, chalo. So let's say you have your mother and you have your father, okay, or male or female. They are going to give you one set of chromosomes each. So let's say mother is giving you one, two, three, four, five, six, dot, 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 22. And then mother, if this is the mother, then she is going to give you the 23rd chromosome, which is going to be only X. Okay, because females have X only. Okay, and now father is also going to contribute to the same chromosomes, right? So chromosome number 1, 2, 3, 4, 5, 6, dot 22, and then you are going to have either X or Y, because males, they give you two chromosomes because males are XY, so they can give you either X or they can give you either Y. Now, guys, what happens? See, I am calling this chromosome number 1. I am also calling this thing chromosome number 1. So there has to be some reason why am I calling this as chromosome 1, right? Why am I calling them chromosome 1? Because both these chromosomes, they are going to be similar to one another, exactly similar to one another. Okay, so that is why they are same. That is why I call them chromosome number 1 from the mother, chromosome number 1 from the father. Same way, guys, I can say I have chromosome number 2 from the mother, chromosome number 2 from the father. They are also going to be the same, right? So they are also going to be homologous chromosomes. Let's say we are going to have chromosome number 3 and chromosome number 3, they are also going to be your homologous chromosome. They are similar to one another, so these are nothing but your homologous chromosomes. Okay, these are all going to be your homologous chromosomes. So what are these homologous chromosomes? Homologous chromosomes, these are going to be your chromosomes. Okay, so I'll just write down the definition out over here. What are homologous chromosomes? Okay, so homologous chromosomes are the chromosomes which are similar in size, which are similar in size, shape, okay, which are similar in size, shape, structure, and genetic material. Size, shape, structure, and genetic material, but obtained from one parent each. But obtained from one parent each, meaning one you are going to get it from the mother, one you are going to get it from the father, and these are nothing but your homologous chromosomes. Everybody getting this thing? So like this, we have your 22 homologous chromosome pairs. Okay, so until here, everybody is your homologous chromosome. Now, you may say, sir, but what about the last one, 23rd pair? See, 23rd pair can keep on varying. Either you can have XX or XY. If you're having XX, if you're a female, then yes, you're going to have homologous chromosome. But if you have X and Y, then they are not going to be exactly alike. So that is not going to be your homologous pair. But in that also, X and Y, they do have some regions which are going to be homologous, some regions which are going to be non-homologous. Okay. However, for the time being, we'll not talk about it because that for that we'll have to go into your actual genetics part. Okay. So, but then that we'll do it a little later on. But for the time being, guys, for the time being, this is what is going to happen. Everybody okay with this? Has everybody understood what is your homologous chromosomes? Okay, in your exams, guys, they always ask you define synapses. Okay, so the reason is because there is another term. Just a moment, guys. I'm getting a call from the classes. 1 minute. Okay, so guys, has everybody understood what are your homologous chromosomes and how is the synapses happening? Guys, with this red chromosome will only pair with red chromosome. Red chromosome cannot pair with your blue chromosome. Okay, get that thing. The color needs to be the same. Okay, they have to be of the same type, guys. Okay, is everybody all right with this thing? Sure. Okay, only Zui is answering. Anybody else? The guys, Sachi. Oh, once again, call from the classes, guys. 1 minute. One minute. 1 minute. They give me blank call right now. Nobody was speaking. Okay. Okay. Anyways, chalo. Let's move ahead, guys. Are once again, 8 seconds. Hello. Hello. Okay, chalo. So, guys, now let's try to move ahead. Okay, so after your leptotene, we have zygotine. After zygotine, we have once again the most important one, that is your pachytene. Guys, this step is very, very important. Very, very important. Okay, chalo. So, let's try to see the next one, pachytene. Okay. Okay, so guys, in patchy, now what does patchy means? Patchy out over here, it means thick. Patchy means thick, and teen means stage, meaning something is going to be thick out over here. Something is going to be thicker, something is going to condense, something is going to become like really, really short and stout. Okay, let's try to see what exactly is happening. So, guys, once again, what are we going to have? We are going to have our cell membrane. The cell membrane is intact as it is. The nuclear membrane is intact as it is. The nucleolus is going to be intact as it is. Okay, until now there is no change in your internal this thing, that is your nuclear membrane, nucleolus. Okay, once again, your centrioles, they are still moving towards the opposite poles, they are going away from one another. Okay, okay, once again you are going to see that, oh, they are still going to be held together by the spindle fibers. Okay, so these things, they are going to remain the same thing. But now, guys, what happens? See, we had in your leptotene, what did we say? See, guys, out over here, we said that, oh, your chromosomes, they were like, very, very thin, they were very, very stout, they were thread like, right? And they were arranged in a zipper like fashion. Now, here in this case, guys, you are going to see that the condensation is going to be complete. The condensation is complete, and now those thread like structures, those are going to become these egg-shaped structures like this. And now when you look at these egg-shaped structures, guys, what you are going to see is that these egg-shaped structures, they are actually overlapping one another. They are going to hold one another like that. Okay, so same way out over here, you are going to see another one like this. They are going to hold one another like this. Okay. Now, let us try to see what exactly is happening out over here. See what has happened. The chromosomes, they have condensed. The chromosomes have condensed and they have become this egg-shaped structures. That is fine. But now, let's try to see. Okay, so now let's assume, guys, we are zooming in into this egg-shaped structures. Okay, these the second one. Now, actually, what happens, I will show you. Okay, now I'm showing you what was happening at the time when they were like, overlapping one another. Okay, right before that and right after that also, I will show you. So, guys, let's assume we have one chromosome, that one blue chromosome, it is going to be like this. It is not shaded. Okay, so this is not shaded. And the other blue chromosome, guys, let's imagine this is going to be shaded chromosome. Okay, so let's imagine this is like completely shaded one. Okay, so first thing, okay, so this is like this. Now, guys, I want you to understand a few terms. Okay, and guys, I need your help also. You need to keep on answering. Okay, so guys, once again, what are we going to say? Guys, say, can I say this and this? They are guys, can you tell me what are they? We have said that one part, if this and this. Very nice. What are these are going to be sister chromatids. Very good, guys. Very nice, very nice. Okay, so we are saying that, oh, these are our sister chromatids, right? Okay, chalo. So if these are sister chromatid, guys, can I say these two, they are also sister chromatids, right? So, okay, these are also sister chromatids of their respective chromosomes. Okay, but now, guys, if I have to tell you this and this, tell me, are they going to be sister chromatids or are they not going to be sister chromatids? Give me yes or no. What do you think? Very good, guys. They are not sister chromatids. Yes. So because these two, now if you have to refer to them, guys, we are going to use the term non-sister chromatids, because these are chromatids from two different chromosomes. That is why these are non-sister chromatids. Okay, everybody getting this thing? Sure. Okay, so see, guys, we are having sister chromatids, and in between, if I want to talk about these two, they will become non-sister chromatids of one another. Okay, fine. But now let's try to see what exactly is going to happen. So at the time of crossing over, guys, you are going to see that these two chromosomes, they are going to come really close to one another. So you are going to see that, oh, we have this one chromosome and we are going to have the other chromosome. But now when these chromosomes, they are going to come together, guys, what happens is these chromosomes, they start overlapping one another, and now you are going to see that, oh, they have like overlapped one another like this. This is what is going to happen. Now, guys, this point, okay, this point where overlapping is taking place, and this entire process, what is this? We will talk about this. So, guys, this, first of all, this point, okay, the point at which the two non-sister chromatids have overlapped, this point is called as your chiasma. This is called as chiasma. Chiasma is singular, chiasmata is plural. Okay, so we have chiasma and we have chiasmata. Okay, so now what is this? If I have to define this chiasma, what is this chiasma? This is nothing but point of intersection. Okay, point of intersection of your non-sister chromatids. Okay, and what is this entire process called? See, what is happening out over here when they are like overlapping one another? This process, we call it as crossing over. The process is called as crossing over. So what is this crossing over? Crossing over is the process in which non-sister chromatids, non-sister chromatids of homologous chromosomes overlap one another. Non-sister chromatids of homologous chromosomes overlap one another. That is nothing but your crossing over. So guys, in your pachytene, what is the most important thing that is going to happen? That is crossing over. And guys, why is this crossing over important? Because this crossing over, ultimately this is going to lead to genetic recombination. It leads to genetic recombination, or this is going to lead to your genetic variations. How it is going to happen, I will just show you. Okay, genetic recombinations or genetic variations. Fine. But now, see guys, okay, chalo, here they have, like, crossed over one another. But what is the end result? What is going to happen at the ultimate end? Guys, during your anaphase one, what is going to happen is both these chromosomes, they are going to separate out. But now, when they are going to separate out, guys, you know what is going to happen? See, the place where they were overlapping one another, right? The place where they were overlapping one another, guys, you are going to see that this part, whatever that was overlapping, that is going to get exchanged. And this is nothing but genetic recombination, guys. And here you will see that, oh, the part, the arm which was out over there, which was like, you know, like this. No, let me just. So in this case, what is going to happen? See, the one which was colored, or the one which was shaded, 1 minute. Okay, its part has gone to the left one, and the left one part, it is now going to go to the right one, guys. See, this is how it is going to happen. And you know what? Guys, this is, I'm showing you, crossing over taking place at only one point, only one chiasma, right? In reality, there are going to be thousands and thousands of chiasmas present in your homologous chromosomes. So each and every place you are going to see that, oh, there is going to be that entire genetic exchange taking place. Okay, once again, just to give you a little bit of idea. Okay, so guys, let's imagine now I'm drawing it like thin thread like only. Okay, so you are going to have one like this, you are going to have another one like this, right? And let's imagine we have the other homologous chromosome. Imagine, okay, now I'm drawing it another color because I want to show it to you what is exactly going to happen. Now when they are going to cross over one another, guys, see, it is going to cross like this. It is not going to be at only one point. You are going to see that, oh, it is crossing over at like hundreds and hundreds of points. And wherever it is happening, guys, wherever this crossing over is taking place, at that place you are going to see this kind of exchange taking place. This is your crossing over, and that is why this is the most important thing which is going to lead to your genetic recombination. Okay, everybody okay with this thing? Yes, getting this part properly, guys. Have you guys understood what has happened? Okay, so if you, okay, if you want, I will once again put down who's going to become thick out over here. The chromosomes. Okay, so the chromosomes, they are going to become thick, and two important things. So you just showed them in the thread like form. Yes, that is just to make you understand what exactly is going to happen. Okay, because see, if I draw, you know, if I draw this kind of a structure, then it becomes difficult to show you. No, you will not understand what is happening. So if I have to show you only one part, crossing over, then I will have to show it to you like this, like this, like this. So see, it doesn't look proper. You won't understand what is happening. Getting it? So I cannot show it to you this way because you will not understand. So I have to show it to you in this fashion. Okay, so always remember, crossing over is very complex. This is very, very complex. Okay, Vaishnavi, all right. Okay,
Sure. So, guys, now after this, what are we going to have? Okay, now after your crossing over has taken place, see this separation, now, guys. 1 minute. There's someone who has messaged me some long thing. This is going to happen. The separation is going to happen in anaphase one. Okay, so anaphase one will separate the two. But I have to show it to you, like, what really happens. 1 minute. There's one person who has messaged me, some big thing. Let me read. Okay, yes, the message is from there. So crossing over will take place only between two chromosomes of the homologous pair, right? Yes, absolutely. Because only the chromosomes which are similar in size, shape, structure, genetic material, only they can show crossing over. So chromosome 1 will undergo crossing over with chromosome 1 only. Chromosome 1 cannot go crossing over with chromosome 10 or chromosome 20. No, it will be with chromosome 1 only. Okay, another chromosome from some other homologous pair cannot show crossing over. No, you know, see, one will show crossing over with one only. One cannot go with two, three, four, five, six. It cannot go with that. It can go with only this. Okay, same way I have two can go with only two cannot show crossing over with one or three or four, five, six later on. So that way, so let's say three is going to show crossing over with only three cannot go with one, two, four, five, six, that way. Okay, everybody all right? This takes place in all the 23 pairs. Now for you guys, for the time being, 22 pairs. 23rd pair, it happens, but it happens partially. Okay, because in your 23rd pair, now see, there are two possibilities. The 23rd pair, see, the 23rd pair, it can be either XX if you're a female. Okay, now if you are having XX and if you're a female, then yes, then crossing over will be seen in all the 23 pairs. Okay, but then in your males, in males, what is going to happen? You're going to have X and you're going to have Y. So both these chromosomes, they are not the same thing, right? But then in your X, let's see once again, if I show you this X chromosome, okay, you will see that little part of X chromosome and little part of Y chromosomes, they are also matching. Okay, so crossing over will only take place in those matching parts, not in the parts which are different. So in males, the 23rd pair is going to be partial, that way. Okay, got it? Okay, chalo, very nice, very nice. Okay, chalo. So guys, has everybody understood what is crossing over? How is it going to happen? What is patching? Okay, after your pachytene, guys, now we are going to move into your diplotene. Okay, so let's try to see what happens in your diplotene or diakhinesis. Diplotene and diakhinesis. Let's try to see. But now let me just see. Okay, we have a little bit time. We will be able to finish diplotene. Okay, diplotene or diplonema. Guys, you need to remember Diplo means two. So two means there is going to be some kind of separation kind of a thing that is going to start out over here. So here in your diploting, guys, what you are going to see, okay, let me just show you. So once again, you are going to have the cell membrane, which is going to be as it is. But now when we come to your nuclear membrane, guys, here you are going to see now that the nuclear membrane, it starts disappearing. It starts disappearing. The nucleolus, it starts disappearing. The nuclear membrane and the nucleolus, they start disappearing. But now, what about the centrioles? Okay, the centrioles, they are trying to reach the opposite poles. That is still fine. Okay, chalo. So they are going to still reach the opposite poles. What happens to the spindle fibers? Once again, they are still going to be held with the help of your spindle fibers. Okay, so they are still going to be held together with the help of spindle fibers. Okay, now after this, what is going to happen, that is very important, guys. See, in your Apache what did we say? See, how were the chromosomes? The chromosomes were attracted to one another and they were overlapping one another, crossing over. Now here in your diplotin, now what happens is the forces of attraction, they are going to end. The forces of attraction end and the forces of repulsion begin. So you are going to see that, oh, these chromosomes, now they are going to start stretching out and they will start moving away from one another. But then still they are going to be held together at the chiasma. But then they will start moving away from one another because the forces of attraction have ended. Okay, so you start moving a little away. This is what is going to happen out over here, guys. In your diplotin, what is going to happen? The forces of attraction between the two chromosomes, they end, and the forces of repulsion begin. Forces of attraction, they end and forces of repulsion begin. And the second thing that you are going to see, that the nuclear membrane and nucleolus, nuclear membrane and the nucleolus, they are now going to start disappearing. They both start disappearing. That's it. That is what is going to happen, guys. Okay, everybody getting this part? Fine. Okay, and now after your diplotin, we are going to have the last phase out over here. And what is that last phase? That is nothing but diakhinesis. Now, dia means what? Across. Kinesis means movement. Okay, so dia, it means across, and kinesis means movement. So there is going to be across movement of something. Okay, what? So guys, you are going to see that, see here, where were the chromosomes? The chromosomes were like overlapping at the chiasma, and somewhere it was in the center, right? So let me just pull this thing out. Let me pull this picture. Okay, so guys, until now, okay, until your diplotin, अरे ये क्या हो गया? Anyways, I'll just draw. Okay, until your diplotin, guys, how was the situation? You were having the chromosome which was like this, okay, and the other chromosome which was there, okay, it was like this, right? So they were like, holding one another, but where exactly? They were somewhere towards the middle part. In your diakinesis, now what is going to happen is you are going to see that that middle portion, whatever that is there, like the chiasma, okay, instead of it being in the center, now it is going to be pushed towards the end. You are going to see that, oh, this is now going to be completely pushed towards the end out over here, like this. Okay, and what is this process? This process is called as terminalization. So in your diakinesis, see, this is across movement of the chiasma. That is why across movement of chiasma. And ultimately, what is this going to result into? It is going to result into terminalization. Sir, can you explain the first payment of diplotin again? Point. forces of attraction are going to end. So see, they were like, earlier, why were they overlapping? They were overlapping because they were being attracted to one another that way for your crossing over. Now what happens? Suddenly the forces of attraction are going to end. So just imagine, okay, just imagine, okay, okay, let's imagine you are holding your partner's hand and you are walking on the road. Okay, so you are hand in hand walking on the road, and now suddenly you happen to see your dad. Are you still going to hold the hands? No, maybe you're going to be scared, and then what? Suddenly, immediately you'll be like, "Bazula, bazula, bazula," right? Why? Because suddenly, instead of forces of attraction, forces of repulsion, they are going to become more predominant. Otherwise, what will happen? One tight slap, right? So this is what is exactly happening. The forces of attraction between the two chromosomes, they are going to end, and suddenly, because of chemical changes out over here, they start repelling one another. That is going to happen. Got it? Rui, are you all right? Yeah. Okay. So here, this point, this is going to be nothing but terminalization. Okay, but now what is this terminalization? This means nothing but the chiasma, it is going to come up at the end. The chiasma reaches the end. Okay, so that is what is going to happen. So guys, once again, if I have to show you once again the cell diagram looks exactly like that. Okay, exactly how we have seen it in that case first. Okay, however, what are the changes that are going to happen, guys? Here the nuclear membrane is going to disappear. The nucleolus is going to disappear. The centrioles have reached to their opposite poles. The spindle fibers, they are going to be present. But now what is going to happen, guys? You are going to see that now the spindle fibers, they are going to start attaching to your chromosomes. But guys, see here, how are the chromosomes? The chromosomes are in pairs. So in this case, the biggest difference in your mitosis and meiosis, in mitosis, you had one chromosome on one spindle. But here, how are you going to see? You are going to see two chromosomes on one spindle fiber. Getting this thing, guys? Why? Because they are still held together with the help of your chiasma. Okay? Everybody all right with this? Okay, so this is the biggest change, and this is nothing but what is your prophase one. So right out over here, the prophase one is going to end. So guys, once again, let me just put down out over here what is the important thing. So first thing, you are going to see that terminalization is going to take place, where the chiasma is now going to be shifted towards the end. Second thing, you are going to see that the nuclear membrane and nucleolus, both of them, they are going to disappear. Both of them, they disappear. Okay, and finally, what are we going to see? The chromosomes. Okay, the chromosomes are going to attach to the spindle fibers. But in what manner, guys? Can you tell me? Are they going to be arranged in some particular order, or they are going to be arranged quickly, like how we had ended prophase and mitosis? The chromosomes are going to be attached to the spindle fibers randomly. Very good, guys. Very nice, very nice. So they are now going to be attached to the spindle fibers randomly. That's it. This is where, guys, our first thing is going to happen. This is our entire prophase one. Okay, so leptotene, zygotene, pachytene, diplotene, and finally diakhinesis. Okay, with this thing, guys, after this, what is going to happen? The cell is going to enter into your metaphase one. Okay, but then that metaphase one onwards, we'll see it in the next class. Okay, all right with this, guys? Sure. Okay, chalo. So, guys, we will stop over here for the day, and I will see you in the next class. Till then, take care. Bye bye, everybody. Bye bye. 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 Bye.